Today I want to tell you about fatty acid breakdown inside of the human body. Specifically, I want to go through the steps of beta oxidation of fatty acids. And even though that name may sound intimidating, what I want to show you after this short lecture is that the steps are very straightforward, and what we can do is better understand how our cells utilize the energy stored in fat molecules to do all sorts of work inside our body. Now, our circulatory system is going to deliver fatty acids to various cells throughout our body. These fatty acids can come from our diet or from reserves within our adipose tissue, but these fatty acids need to enter the cell where they undergo an activation that's an ATP-dependent process and eventually get transported into the mitochondrial matrix, which is an active process. And it's here that they'll be broken down as fuel. Now, a fatty acid that is activated will be covalently attached to coenzyme A. And this process allows for the head to position the alpha and beta carbons of the hydrocarbon tail of the fatty acid specifically within the active site of various enzymes so that biochemistry can happen in this region. And so in the next slide, I want you to focus on this region of the alpha and beta carbons to see the changes that take place throughout this pathway. If we begin with lauryl-CoA, which is a 12-carbon saturated fatty acid inside the mitochondrial matrix, this particular molecule will react with FAD in the enzyme acyl-CoA dehydrogenase. This enzyme is, as a dehydrogenase, going to do redox chemistry. And for this first step, we're going to reduce FAD to form FADH2, where this molecule picks up 2 electrons and 2 protons from our lauryl-CoA substrate that gets oxidized to form this enol-CoA intermediate. And as you can see, the alpha carbon and the beta carbon form a double bond between them and have lost a proton each. These protons are the protons that went to FADH2. And this FADH2 is going to provide energy for the cell. So this is the first reaction of beta oxidation. Our second reaction is catalyzed by an enzyme called enol-CoA hydratase. And as the name implies, this enzyme, a hydratase, is going to do a hydration reaction where we're going to split a water molecule into a proton and a hydroxide group. We're going to put the proton on the alpha carbon, and as you see, we put the hydroxyl group on our beta carbon. This generates the beta hydroxyacyl-CoA intermediate that is the completed step of the second step in beta oxidation. Our third reaction is another redox reaction that's catalyzed by beta hydroxyacyl-CoA dehydrogenase. Instead of using FAD, this dehydrogenase uses the cofactor NAD, nicotinamide adenine dinucleotide, to transfer 2 electrons and 2 protons from this beta carbon to one of them going to NADH and the other proton being released into the medium. This reduction of NADH is coupled to the oxidation of the substrate fatty acyl-CoA to generate this keto-acyl coenzyme A, which, as you see now, our beta carbon forms this carbonyl group. And this reaction, this third step of the pathway, is where the entire pathway gets its name. As you see, we are oxidizing or losing electrons from the beta carbon. So this is why it's called beta oxidation of fatty acids. Our fourth step is going to be catalyzed by an enzyme called thiolase. And what we're going to do in this reaction is cleave this keto-acyl fatty acid into 2 products. One of the products is going to be acetyl-CoA, which is going to be this part of the molecule, and we're going to cleave that alpha-beta bond to generate a fatty acid attached to a second coenzyme A. This fatty acyl-CoA can now undergo the next round of beta oxidation because it's simply 2 carbons shorter than our initial substrate. And this continues, the 4 steps of the beta oxidation cycle continues until, in the very end, we're going to generate all of the acetyl-CoA molecules we can, and we're going to generate FADH2 and NADH that are going to be high energy intermediates to give us ATP equivalents through oxidative phosphorylation. Now, where do all these ATPs come from? Let me just simply walk you through this very quickly. Lauryl-CoA with 12 carbons is going to generate 6 acetyl-CoA molecules. Again, 2 of these carbons from acetyl-CoA are going to derive from the lauryl-CoA fatty acid degradation. These 6 acetyl-CoAs are going to give us a total of 60 ATPs, and these 6 molecules of acetyl-CoA are produced through 5 cycles of beta oxidation. And from these 5 cycles, we get 5 FADH2 molecules and 5 NADH molecules, which will give us another 20 ATPs. And so if we add these up, we get 80 ATPs gross out of our beta oxidation pathway. But as I mentioned at the beginning of this lecture, the activation process requires 2 ATP equivalents to get this particular fatty acid activated and ready for catalysis. And so we have to subtract those ATP molecules, which gives us a total of 78 ATPs per 12 carbon fatty acid. If you remember, glucose, which has 6 carbons, only gives us about 30 ATPs. So you can see that this single molecule fatty acid, even a medium chain fatty acid like lauryl-CoA, is actually giving us over twice as much energy as glucose in carbohydrate metabolism. And so this breakdown process of taking fatty acids and turning these into intermediate products that then can go through the citric acid cycle to be fully oxidized to carbon dioxide, coupling all of these pathways together with oxidative phosphorylation will give the cell tons of energy. And this energy can be used by the cell to do work and keep our body warm.